So first, just a reminder that you can drop in tomorrow and Friday. I'll be in my office for sure, 10 to 11. Might be in my office most of the day tomorrow. Friday, can't remember what I'm doing on Friday, but you can drop in or make an appointment, and feel free just to pop in or let me know if you're coming. I can do Zoom appointments as well if anyone wants to meet that way. That's another option. Okay, couple of reminders about the exam. Don't forget to arrive early, bring your photo ID. I think we've gone through all this already, but it's just good to throw those reminders out. So generally, the way that I do these review sessions is I have all these study questions, and what I'm hoping is that people will have some questions, right, that they want to go over. Usually what I do is show you the study questions at the beginning. I'm just going to flip through these, and I have a few that I'm going to pick on. That'll probably take me I don't know, half an hour-ish, and then we'll pick on any of the questions that that you're interested in going over. So we'll do that. Here's topic on disease transmission and epidemiology, and lots of things to think about there. Next topic on microbiomes and mechanisms of pathogenicity and all of those kind of things. The immune system was broken into 3 sections, one on innate immunity, adaptive immunity, and immune disorders and AIDS and HIV. And then lastly, we covered, kind of reviewed, but covered a bunch of infections. And so it was a little bit review, a little bit new. And so lots of things to think about in there. So I'll show you some of the questions that I want to pick on. And like I said, feel free to ask any questions in the chat or with your microphone on, doesn't matter. Either of those work for me. And once we're done going through those, you can we can see which questions people want to discuss the most. And like I said, this is your review session, so hopefully you know what areas you want to pick my brain on and try to sort out some details on. And so what I've done going through these questions is I've tried to pick on some important concepts, but also at the same time also pick on maybe reorganize things just a little differently. I know sometimes people respond differently when things are organized in different manners. So here's one to start off with, right? What 4 factors contribute to nosocomial infections? So if you don't know, first, what is a nosocomial infection? This is something that's been caught in a hospital or healthcare setting. They used to be called hospital-acquired infections, but you're starting to see everywhere is defining as healthcare-acquired infection because that just makes sense. There's lots of places that are not hospitals, clinics, nursing homes, you name it, that are healthcare acquired and really do fit under this umbrella. So this is just reiterating the slide. We had basically 4 things, right? So it is a healthcare setting. There is a higher chance that there may be pathogens there that are infecting someone else, and those people are going to be acting as reservoirs. Some of these microorganisms may in fact be super bugs or drug resistant, so that's a big deal, of course. Number 2, people who are in healthcare settings may be immunocompromised. They may be elderly or have some other complication that they're dealing with that's going to suppress their immune system, make them weaker and a more susceptible host, really. Number 3, we have all the people there. So hospital equipment, staff, visitors, all those things could be vectors, meaning that's how the disease, the pathogen is getting from one person to another. And then lastly, invasive procedures, catheters kind of being the number 1. I can't remember what the stat is on that, but at least a third, possibly closer to half of these infections are from catheters. Again, you know, the numbers don't matter so much. It's just know that catheters are kind of the number 1 nosocomial infection. Okay, so that was an easy one. I wanted to just start off with something relatively straightforward to break the ice. I'm gonna, for a lot of these other ones, I'm actually gonna be writing on the screen, trying out my digital pen. We'll see how that works. Okay, so number 2 question says, what are 4 sources of pathogens? So I think I mentioned this in lecture that I had given this, I think it was last year, this question. And by sources, I meant reservoir. So some people were giving me some really weird answers on this. And I think they were confused with mechanisms of transmission. So some people were talking about direct contact or sexual contact or soils. Actually, soils is a source, a reservoir. But mode of transmission is a different question, right? And it's all kind of connected to this, cycle of infection, of which all these are things we talk about. You can see reservoir is one of them. So this is a good question because I can ask you what they are, and I can ask you examples as well, right, of different reservoirs and different microorganisms coming from these reservoirs. So number 1 is your normal flora, and lots of examples for that one. I don't know if I have space at the bottom, so I'll put some examples up here. So E. coli is a good example. E. coli, of course, is found in your intestine. It's part of your normal flora. Can cause yeast or not yeast infections, bladder infections, and other opportunistic infections. Staphylococcus is a good one. So both Staphylococcus aureus and Staphylococcus epidermidis. This list could go on. We could talk about Neisseria meningitidis causes meningitis. Many of us have that on us as part of our normal flora. Often streptococcal organisms as well can be found in the saliva and other things, and sometimes get into other body parts and can cause infections. But E. coli and Staphylococcus aureus are a couple of good examples on that. Number 2, human carriers. This is probably the most obvious one. Somebody gets sick and they give it to you, and it could be a variety of ways. And this covers many of the diseases, probably the majority of the diseases we talk about in this course. So COVID-19 is a good example, which originally was a zoonotic disease, but now who's catching it from animals? Nobody, none of us. Most of us are catching it from other people. This would include, of course, all of the sexually transmitted infections. So that would be gonorrhea, chlamydia, and so on, and many other types of infections, of course. And sometimes we could be looking at organisms that are transmitted fecal orally if humans are the reservoir. So certain types of pathogenic E. coli would be a good example of this. So animals, remember that means zoonotic. And so rabies is the classic example of that. And that nobody is getting rabies from another human. I don't know if that has ever happened. I have no idea. I guess it's possible, but usually it's from direct contact with animals. Certain types of flu. So for example, we talked about avian flu. Obviously, avian means bird. So that means it's coming from contact with a bird, usually the feces. Again, many we could put on this list of organisms that fall into that category. Those are a couple of good ones, though. Sometimes you see the tapeworms falling on this list. So the beef tapeworm and the pork tapeworm, which are not usually due to animal contact, and humans are kind of the reservoir as well. It's because they're part of the life cycle. So that's kind of a gray zone, but I would accept that as a correct answer because animals are involved in the transmission and essential for it. And then lastly, abiotic. So we're talking about organisms that can live naturally in the soil and water. So in this case, we're talking about, for example, tetanus, which people associate with rusty nails for some reason, but the rust has nothing to do with it. It's more of the dirt, because rusty nails can be dirty and the microorganism can live in there. Botulism as well is another good one. And both of them are Clostridium organisms that are endospore forming, and they live in the soil. So there's a couple of good examples that are common enough in Canada. Cholera would fit in that category as well. We haven't really talked about cholera too much in this semester, but it can live naturally in water sources, and so that fits into that category. Okay. Here's another one. Speaking of water sources, something I maybe kind of missed there in the last topic. We talked about waterborne infections and all sorts of gastrointestinal infections that are spread by fecal matter. But there's one waterborne infection that kind of is just unique. It's an exception from all these other waterborne infections, right? And so it has a different cause. Legionellosis is from Legionella. So this is the cause. We'll put there. And so that's a unique organism, right? So that's why it's called Legionellosis. Other waterborne infections, right? So we have pathogenic E. coli. What else do we have? I'm just trying to rack my brain to remember what we're talking about. Hepatitis A. Hepatitis A. There we go. Got to spell correctly today. What else do we talk about? Norovirus. A whole bunch of them. Sometimes you see salmonella in this. Salmonella can be waterborne. It's more likely to be foodborne, but I guess we could throw that down here. What else did we talk about? Giardia. Polio. This list goes on and on and on. There's a bunch of organisms I can think about that we didn't really talk about too much. Shigella. What else am I thinking about? Some of the schistosomes and things like that. But I think that's a good list for now. Okay, so what else is different that we can compare? Secondly, we can talk about transmission. So we'll put that down here. Transmission. So Legionella is an environmental organism, and it likes to live in warm places. So anywhere where humans have warm water. And if you remember, we were talking about kind of one of the, there's two big scenarios where we see Legionella. One is hot tubs, and two is on large buildings that have these air conditioner units that they have these cooling towers on the roof. And the thing that's in common with both of these is that the bacteria is growing in the water and is spread usually by mists. So it gets aerosolized, and people are going to inhale it and get it in their lungs, right? Other waterborne infections usually has to do with fecal contamination, right? So fecal contamination. Fecal contamination. We could say food and water. We'll say that, of water and food. And it could be animal feces, it could be human feces. It just kind of depends on the organism and the circumstances. So last one I'm going to put here on this list is symptoms. And so if Legionella is inhaled into the lungs, we're not going to usually get any gastrointestinal symptoms. It's all going to be pneumonia, right? So sometimes this is called Pontiac fever. So there's a fever often associated with this as well. So we'll call this pneumonia. Or you could call it a pneumonia-like disease. I don't know why you see that in the terminology. It's basically a type of pneumonia, but caused by a specific bacterium, of course, and a fever. So other waterborne infections, what are we talking about here? Diarrhea and vomiting and other gastrointestinal kind of symptoms. Diarrhea, vomiting. Here we go. So that's kind of the basic comparison. And so some things to think about. Like I said, most of these waterborne infections are kind of similar in manner, right? You've got the fecal contamination, human or animal, diarrhea, vomiting, intestinal cramps, sometimes fever, sometimes blood in the diarrhea, but a lot of common ailments. Legionella, which is actually a little bit more common in Canada because, hey, we have hot tubs and some of those people don't clean them out so well. And so that can be a source of the bacterium growing in the hot tub. Okay, what do we have next? Okay, here's a good one. So it says compare and contrast the causes, reservoirs, modes of transmission, symptoms, and treatments of Lyme disease and West Nile fever. All right, so made a little table for that. And so let's fill that in. And maybe what we'll do is we'll just focus on Lyme disease first, and then we'll do West Nile virus. So remember, Lyme disease is caused by a bacterium, and it has a name. It is called Borrelia burgdorferi, if I'm saying that right. So Borrelia, did I spell that right? I think I've got a note here. I think there's actually only one L. Let me see if I can erase that. Here we go. And burgdorferi. So burgdorferi is named after a guy who studied it. Interesting name. And so, I'm going to draw a little bacterium here. It's a spirochete. So a twisty bacteria. There it is. Looks something like that. Okay, and what's the reservoir for Lyme disease? The Lyme disease is a mouse. It's actually called the main reservoir anyway. It can live in other animals, but the white-footed mouse seems to be the most important. There are some areas in Canada where this has been looked at, and something like 80–90% of the mice seem to have the bacteria in them. Some people think the deer. The deer can be important for the tick, but the deer don't usually have the bacteria, maybe up to 10% in some populations. And after the tick bites the deer, it's not usually likely to bite the human. So the mouse is the most important part of the reservoir. The mice and the deer have very similar habitats, and so there's a lot of ecology going on in there too. So what's transmission? These are both vector-borne diseases, and so this is usually called the black-legged tick, which these ticks here do have scientific names, which I can never remember the names, and that's the most common name for the tick. Sometimes I've seen them called deer tick. And if you remember, there's actually two versions of this tick. There's a version that's called the Western black-legged tick, which is actually a different species, and it's found west of the Rockies, so a small part of Canada. What are the symptoms? The big one to remember for Lyme disease is the bullseye rash. Bullseye rash. So it looks like a bullseye on the skin, usually found around lymph nodes. And this is about 80% of people get this, we think anyway. There might be a few less, at least symptomatic people. This is what we see. Sometimes there's fever and all those typical kind of things, but another big one is arthritis-like symptoms. Arthritis-like symptoms. Fatigue is a big one, and lots of, there's a huge list of symptoms, but those are kind of a couple of the big ones. The bullseye rash is the big one to look for because it's very unique. There's nothing else like it that causes that rash, at least in North America. And, it's like if you have that rash, you have Lyme disease. How do we treat this? Well, it's a bacterial disease, so we can treat this with antibiotics or antibacterial drugs, we'll say. Now, this is a simplified version of Lyme disease. Some people don't get treated right away, and there's symptoms that they may need to get treated in the long run, but that don't include antibiotics, but we're not going to get into all those details here. Okay, West Nile fever. This is a virus, so this is caused by West Nile virus. So already we're looking at something different here. Sometimes this is abbreviated to WNV. You see that in the scientific literature, West Nile virus. So who's the reservoir in this case? Birds, many species of birds, in fact. Robins, I believe, are one of the big ones, but many, many species of birds. When it first came to Canada, crows were dying. Crows are not the best reservoir for this virus, but there are many, many species that act as reservoirs. In this case here, the transmission is a mosquito. It's actually called the Culex mosquito. You don't need to know the different types of mosquitoes. Culex mosquitoes. There's actually two types. There's sort of one that hangs around in Eastern Canada. I think it's called, a common name is like the household mosquito or something like that. And the one in the West, I can't remember what it's called now, the common name, but it tends to like agricultural places. So, it'll breed in hoof prints and little swamps and ditches and things like that around farms. What are the symptoms for West Nile? So flu-like symptoms. So what do we mean by flu-like symptoms? So fever, chills, fatigue, those kind of things, not necessarily sore throat. And the one that we're most concerned about is encephalitis. So encephalitis. Some people say encephalitis. I think it depends on what side of the ocean you are on. But that's swelling of the brain. And that's where it can be fatal, particularly for older people. In this case here, no antibiotics for treatment. And we treat the symptoms. So if there's a risk of or suspecting of encephalitis, they're going to give the person some sort of steroids to reduce the swelling and until basically the body is going to mount an immune response and clear the virus eventually. Okay, so moving on. I guess I didn't draw any West Nile viruses. I'm not even sure what they look like, but we'll just draw some little dots there. I like pictures. They kind of help me remember things. Okay, so here's another one. It says here we have patient, and the patient has an E. coli infection. It doesn't say what kind of infection, possibly a blood infection. It says, explain why this microorganism, which normally lives in the colon, could kill this patient. So this is a good kind of short answer kind of question. So what are we looking at here? This is your classic opportunistic type infection, right? So what does that mean? That means a pathogen doesn't normally cause disease in its normal niche, but there's some situation that allows that pathogen to able to cause some sort of pathogenesis, right? So lots of examples we talked about in course. I think I kind of break it down into 4 categories. I know there's a nice table in the textbook that gives like, I think, 15 reasons, but again, 4 categories. So weakened immune system. Why might you have a weakened immune system? It could be AIDS. It could be you are elderly. It could mean you're sick from something else, malnourished, etc. Altered microbiome. So you've taken antibiotics and it's messed up with your normal flora, or something like that. In this case, it's category number 3, which means your organism, it goes from one site to another. In this case here, it actually gives you the information here. It says there's an intestinal puncture, right? So that is going to be the opportunity to introduce that pathogen, and well, in this case, a mutualistic organism now gets introduced to another part of the body and becomes pathogenic, unfortunately. Other complications, just kind of a list for other things. There's lots of things out there that may affect somebody's ability to fight off or get disease. For example, cystic fibrosis is a good example. In cystic fibrosis, these people have kind of a thick mucus in their lungs, and so they're more susceptible to lung infections, particularly from Pseudomonas. Other complications, there's probably lots of things we're not necessarily thinking about, but for example, somebody could be pregnant, and sometimes that changes your pH chemistries and those kind of things. And you are, as you're pregnant, you're actually suppressing your immune system a little bit to try to not reject the fetus. So sometimes pregnant people get infections or other conditions they don't normally get. Okay, so you're probably wondering, what do you need to know about pathogenic E. coli? So here it is. This is the list I made for you. There's many types of pathogenic E. coli, but this is kind of the definitive list for this course, right? So first of all, some E. coli can cause urinary tract infections. They're usually called uropathogenic E. coli. And why can they do this? For two reasons. One, they are getting introduced to a new part on the body. So from the colon or rectum to the urethra or deeper. And secondly, that they have virulence factors such as these pili. These pili allow them to attach to the tissue inside the urethra and the urogenital tract. There are many types of pathogenic E. coli. They're usually causing some sort of diarrhea, and they also often have virulence factors such as enterotoxins and things like that that are allowing them to cause that disease. And most of them are spread by human fecal matter. The exception is E. coli 0157. Now, that's not a 0, that's an O for the letter O. You can ask me about it if you want some time. This is the one that causes bloody diarrhea, so dysentery, and it comes from cows. So that's kind of the one to know a little bit more about, because it's kind of a unique category. And as I mentioned before, that's the one that is usually making the headlines and causing people to get quite sick. Okay, I guess I said that already. They're pathogenic because they have virulence factors such as enterotoxins and pili. And the last note there is that E. coli can cause other opportunistic infections. As you saw from topic 14, there is E. coli pneumonia. There's E. coli can cause blood infections, and there's quite a variety of things it can do if it gets into the wrong place. And that's kind of the key list of what to know about pathogenic E. coli. Okay, moving on. We didn't really talk about cholera, but I can give you a note on this here anyway. And so a couple of things to know about these. These are all different organisms. These toxins are produced by all different organisms. Botulism, of course, is Clostridium. Clostridium botulinum. Tetanus is also a Clostridium. Tetani. And so both of these are soil organisms and spore formers. So there's my little drawing of some of these. Sometimes they look a little bit more like tennis rackets because they're kind of a little fatter around the spore, but they're both rod-shaped and anaerobic spore formers. Cholera is from an organism called Vibrio cholera. Sometimes I talk about it during the semester, sometimes I don't. Didn't really get around to it this semester. But it is an example of a Vibrio-shaped organism, so that's kind of like a bent rod. So it kind of looks something like that. There's my attempt to draw a bent rod. So what about the toxin? Right, so remember that botulism, right? That one is Botox. That's the name brand for people who sell the toxin, and it causes paralysis. Tetanus, sometimes is called lockjaw, and it kind of does the opposite. It causes muscles to lock, so they can't relax. Cholera, this is an example of an enterotoxin. So what does that mean? It, of course, means diarrhea. Okay, 1 R or 2 R's in diarrhea. For some reason, my brain can't remember that. I feel like I could spell a few years ago. For some reason, the spelling part of my brain seems to be disappearing. So it's good to know a few examples of toxins. I have given a question on the final exam that, said something like list 3 different toxins that we discussed in the course and what do they do. So there's an example of 3 of them. Okay, so we're kind of getting down to the last part here. I wanted to spend a little time talking about the two different types of adaptive immunity. And so here's a very open-end kind of study question. And again, I've made a table for us so we can try to compare the two types of immunity. So humoral immunity, these of course are B cells or B lymphocytes. I don't know what I'm doing with this digital pen. I think I'm touching a button on it. Keeps messing me up. And cell-mediated immunity, of course, is T cells or T lymphocytes. And their names, the B and the T, of course, come from B for bone marrow. And T is, of course, for thymus. So what do we mean by mature? By mature, we're talking about clonal deletion. And what do we mean by clonal deletion? It means we're going to eliminate cells that are self-reactive. So that means we are trying to get rid of any cells that may recognize human tissue. We need to eliminate them, which means we're killing off those cells before they're allowed to escape out into the blood and into the lymphatic system and all that. And that's what mature means, right? We're getting rid of all those self-reactive ones. If we don't, it could lead to autoimmune diseases and things like that, which we don't want. All right, next one, it says the type of pathogen or substance or molecule that's recognized, right? So I think for these, for the humoral immunity, I called them freely circulating, right? So we'll say that, freely circulating. Okay, and so what did that include? So that includes, of course, freely circulating viruses before they've infected a cell. So if they're in the blood or whatever, right? Bacteria. And we'll throw in with bacteria and other small foreign cells, we'll say. So that could include yeast and certain protists if they're small enough to be killed by phagocytosis. And last one, of course, is large molecules. And by that, usually we're referring to protein toxins. So, for example, we talked about the diphtheria toxin or the pertussis toxin or the tetanus toxin. Those are all part of the vaccines that we have for these things. So for the cell-mediated immunity, we are talking about cellular things, right? So we'll call these, I'm just trying to think of how to write this. We'll say cellular, let's say cells containing antigens, we'll say. So what do we mean by that? Well, we mean, for example, tumors. Your immune system can clean up tumors. Large parasites. So that could include worms or anything that's too large to be destroyed by the humoral immunity. So it could be worms, protists, all sorts of things. What else? Oh, right, of course, virus, cells that are infected by viruses. So virally infected cells. And I'll throw one more in there, which is, of course, foreign substance, which is transplant tissues, which is why sometimes we have to take immunosuppressants if you have a tissue transplant. Okay, so that's the first part, right? Just thinking about the B cells and T cells and what the targets are. A little bit more. I'm just going to leave that here for a moment and say a couple of things, and then I'll go to the next screen. There are a few more things to compare. So if you have a page of paper, you want to leave some space at the bottom or grab another page if you're running out of space. There is a lot of crosstalk between these things. As I mentioned before, the T helper cells is involved in the activation, and there's immune molecules such as cytokines that help prime these things. But generally, they have their roles here, and they can do their roles very well. Okay, so next it says what happens when the cells are activated. So which cells? They're B cells. So in the B cells, of course, they secrete antibodies. Okay, and that's supposed to mean and. Let me just make sure. Let me erase that. Oh, this pen is lots of fun. Okay, and there we go. And make memory cells. Okay, so what about the T cells? They don't secrete antibodies. T cells become cytotoxic. Okay. Spelling is not my thing today. There we go, that's better. Cytotoxic. Plus, they make memory cells. Plus, make memory cells. Okay, so what does that mean? That's kind of the next question. It says, how is the pathogen killed, right? So the antibodies, that's where we're picking up next here. The antibodies bind the pathogen. So they bind the pathogen, and this usually leads to phagocytosis. For the T cells, they become cytotoxic. So we can say this, cytotoxic cells. They will kill the pathogen directly. And there's a couple different mechanisms, at least, that they do this, but usually what that does is they will inject toxins or something like that into the pathogen. So you'll say example, inject, should be an I there. I'll erase that. There we go. Inject toxins into target cell. Okay, you can see I have one more space down there at the bottom. And it says here, how does the immune system remember the pathogen? And I think we know the answer to that. Memory cells. We'll say long lived. So long live the memory cells. So these, of course, are memory B cells. And these, of course, are memory T cells. So I want to get back to those antigens here in a moment. So just give me a minute on that. I think I've got a slide for that. So yeah, there's lots more we could say about these two types of immunity, but these are the things I want you to focus on for this course, right? So the targets, the cell types, and ultimately, what is going on there? How does the pathogen eventually get killed by the immune system? And so the answer depends on what's going on, either antibodies or whether you have cytotoxic cells. So let's talk about those antibodies. What are the functions of the antibodies? And this is just that slide I showed you in lecture. So what do they do? Number one, opsonization. What does that mean? It means the process of getting phagocytized. I think that's the word, phagocytized. So that leads to phagocytosis. Sometimes this leads to recruitment of those natural killer cells. Those natural killer cells are cytotoxic. So you can see in that image, they're injecting some sort of toxin that punches holes in the membranes of the target cell. So cell lysis or cell death, right? The complement system can be activated, and the complement system can lead to cytotoxic events or can lead to phagocytosis. So it's kind of just helping to amplify and enhance that immune response, the complement system. I know we kind of just really scratched the surface of the complement system on the exam, and I'm not going to ask you anything big about it. Maybe like one true and false or something like that about it if I put it on the exam. Agglutination, so this causes the cells to basically clump up, and when you've got a larger target, this can enhance phagocytosis. And then lastly, neutralization. So neutralization means you're binding antigens on the surface of that thing, and it may not actually be able to carry out its pathogenesis. So if this is diphtheria toxin, our immune system can neutralize it so that the diphtheria toxin doesn't even have an effect on the body. Often this does also lead to phagocytosis. So those are the jobs of antibodies, right? So again, this is a great question, right? On a final exam, maybe ask you for, describe 3 ways that antibodies lead to the destruction of a pathogen or something like that. Okay, couple more things, and then I'll open it up to questions. It says, with examples, describe the 6 types of vaccines. Okay, so let's talk about those, and I'll use, try to define them in minimal words, right? So attenuated, right? This means weakened. So sometimes we call these vaccines live attenuated to emphasize that they are biologically active, but not usually capable of causing disease, but weakened enough that they can grow. Lots of examples of those. The vaccines that are found in the MMVR vaccine, so that's mumps, measles, varicella, rubella. All of those are attenuated strains of the viruses. We also mentioned the flu mist as well. Flu mist. As another example, I'm just trying to think what else there was. There was the oral polio vaccine as well. And so all of those are examples of attenuated vaccines. And that's, one of the oldest types of vaccines that are out there, along with number 2, the inactivated. So inactivated, in this case here, we mean we've killed the pathogen, right? But it's still hopefully going to give us an immune response. Lots of examples of those. There's the flu shot, of course, is a good example of that. Have to look it up. I think the rabies vaccine is killed. There's a few less of these, but another example, of course, is, I guess I meant to put arrows for all of these. The injectable polio vaccine. So that's the one that most of us are getting in Canada nowadays. So the conjugate or subunit vaccines, these are, I didn't really go into the differences between conjugate and subunit. So you can sort of focus on that word subunit. And so this means a part of it, right? So part of the pathogen, we'll say. And examples I gave you of that were the pneumonia vaccines, right? So that was carbohydrates from Streptococcus pneumonia. And I think the recent one has 23 different carbohydrates in it. We also talked about another example is meningitis. So this is Neisseria meningitidis. I'm not exactly sure what subunit it is, but I think it's one of the a protein found on the bacterial surface. I'd have to look that one up. I'm just looking at my notes here, and there's something I can't read in my own handwriting. It might say hepatitis A, but I'm not sure. I have to look that one up. Can't read my own handwriting sometimes. That's unfortunate. Toxoid. So what is a toxoid? A toxoid is an inactivated toxin. So I'd have to look this up, but I think it's usually done chemically. But nowadays, of course, we have genetic ways of doing these kind of things. So I'm not exactly sure what is done. 3 examples of that. Tetanus. Did I just spell tetanus wrong? Yes, I did. Add an extra T in there. Let me erase that. There we go. Or an extra N there. Come on. Eraser, where is that eraser? There we go. Much better. So tetanus, diphtheria. Feel like I've just spelled diphtheria wrong. I'd have to look that one up. And there is a pertussis. There's a couple of different pertussis vaccines. One of them is a toxoid vaccine for the toxin. And then the 2 new ones, these are brand new as of 2020 or 2021. I can't remember when exactly they came out. The whole pandemic's been a blur. But these are examples are for COVID. So the nucleic acid vaccines, usually we're referring to the messenger RNA vaccines. So messenger RNA coding for a spike protein. Codes for spike protein. And the examples in Canada are the Pfizer and Moderna vaccines, both of which now have names. I can never remember. One of those Comirnaty, the other might be Spikevax, but everyone remembers the companies. The viral vector is kind of a similar idea. In this case here, we have DNA and it's delivered by a virus. And usually that virus is something that is not going to replicate in humans. It is something that's weakened or otherwise. There's a few different ways to do this. So maybe you could say harmless virus. And the examples of that are the AstraZeneca. And what's the other one? Johnson and Johnson. Johnson and Johnson. Which I'm not even sure if both of those are available in Alberta anymore. I have to look it up. AstraZeneca is a UK company, and I think we had a contract. And then when it was done, we still have contracts with these other companies, so we don't need as many vaccines as there are out there. And this one had a few more side effects with blood clotting than some of the other ones, so there were some concerns. And when you have the messenger RNA vaccines, why not just go with it, right? Okay, so I almost have everything I wanted to talk about, and then we'll open up for questions. I guess I've gone a lot longer than I thought I was going to. I guess every year I add a little bit more to this, and it adds up. So this says, describe 2 types of immunodeficiencies, right? So remember, immunodeficiencies, focus on the word deficiency. It means that the immune system is not functioning properly. So number 1, we're the primary. And this means you're born with it. And the example that we gave you in class was the SCID. Was that severe combined immunodeficiency disorder or something like that? That's the bubble boy disease, the one where that boy was living in a sheltered bubble to try to keep him away from pathogens. Number 2 is acquired. So by acquired, we mean you're getting it later in life, right? So this could be from malnourishment. This could be from an infection. I'm going to give you 2 examples of infections that can lead to this, right? Measles is a good example. And of course, HIV is the classic example, something that is giving you an acquired immunodeficiency. Remember, hypersensitivities are something entirely different. Okay, so let's just fix that handwriting and the spelling. Sensitivity, there we go. So something different, meaning the immune system is overreacting rather than underreacting. There were lots of examples of these. We had the allergic diseases. So allergic diseases is a whole bunch of diseases that include things like hay fever, allergies, asthma, anaphylaxis, eczema, just to name a few. And we also talked about a bunch of autoimmune diseases. And in this case, like I said, you don't need to know too many particulars. Just know if something is a hypersensitivity or an immunodeficiency. So autoimmune diseases, we talked about like type 1 diabetes, what else? Lupus, rheumatoid arthritis, farmer's lung. There's a whole bunch of these. See, there's a question. Let me see. It says here, someone's just asked me to go back to the slide about the 6 vaccines quickly. Yeah, I can do that. I'll do that in a moment. So take a look, like I said, at the notes on these and notes on examples of these hypersensitivities, and focus on HIV and AIDS, of course. So there's the slide with the vaccines, and I want to say a couple of things about HIV and AIDS, and then, like I said, I'll open it up to questions. I have slides with the study questions, so people can certainly ask me to go over any of the questions you see on the slides. Okay, so let's talk about AIDS and the progression of AIDS. Okay, so we're talking about initially there's this acute phase. So, oops, getting ahead of myself. I was just going to circle this with the pencil or the pen. There's the acute phase. So the acute phase, you can see on those notes there, it's a mono-like illness. So when we say mono, often that's an infection of the lymph. So you're looking at swollen lymph nodes and sore throat, fever, sometimes some weight loss. Sometimes people get diarrhea. There's a whole list of things that people might get. Usually it doesn't seem that serious, although can be. And then people usually recover at that point. So if you take a look, the thing I want to show you is this blue level here. So this blue thing here, this is the CD4 cell. So those are those T helper cells. And you can see that there is an immune response, but those cells are slowly getting killed off over the years with the infection. Red is the viral load. So you can see it starts off and spikes really quickly. And so the infectious stage is often, most infectious stage is often early on in the person's infection. Moving on, and there's the symptomatic clinical phase right here. So this is where the immune system right down here is kind of baselining. And usually, we're looking at clinical AIDS is defined as less than 200 CD4 cells per microliter. Sometimes you see in the literature, they talk about kind of pre-AIDS or AIDS-related complex. So that's where the immune system is beaten down, but hasn't quite hit that critical level. And so you do see some different clinical progression in terms of the types of infections people might get at those different stages. I'm not really wanting you to focus on that too much in terms of which infections you're getting at different stages. And there's a whole bunch of them. I was going to list some of these infections, opportunistic infections that are associated with AIDS or clinical AIDS or pre-AIDS, kind of in that last stage. So we talked about shingles. So shingles, of course, is caused by the varicella virus that people might have had as a child as chickenpox, and the immune system is suppressed. Herpes, so sometimes we talk about with AIDS patients weird herpes infections. So sometimes they get disseminated herpes, which can mean it's rashes in weird places on the body, sometimes all over the body. Sometimes that's also associated with shingles, that term disseminated herpes, because it's caused by a herpes virus, shingles, of course. All sorts of cancers and sarcomas. Sarcoma, that's a cancer of the blood vessels, I believe. All sorts of weird fungal lung infections. Just trying to remember what else we talked about. Toxoplasma. So the disease is toxoplasmosis. And again, we don't know what percentage of Canadians are infected. I saw one estimate at 15%. I saw another estimate at 40%. So we're harboring the pathogen, and it's just ready to pop out when we're immunocompromised. Most of us, it never will. But for AIDS patients, it's very likely to. And the last one I had mentioned was tuberculosis, which is not usually in Canada associated with it, but in many other parts of the world, someone with tuberculosis, often it means they do have HIV infection because they had latent TB, and now it's come out of its dormancy. Okay, so I guess I blabbed a lot. That's okay. I have one or two more slides, and then we'll open up to questions. And I don't have a very busy afternoon, so if this goes 2 hours, I'm totally fine with that. This one here, I wanted to I kind of put together, a list of types of infections caused by staph, right? So we did talk about, of course, our skin infections. So a whole bunch of skin infections we talked about, but don't forget it can cause other infections, respiratory infections, urinary tract infections, food infections, nervous system infections, systemic infections, you name it. Very, very good at infecting multiple body systems. And I put this slide up here because I think last year or the year before, I put on one of the exams, name 3 infections caused by Staphylococcus that are not skin infections, and people were giving me skin infections. So if I ask that question, just be warned. Don't give me skin infections if I say don't give me skin infections, please. What else do I have here? Okay, so I guess that's it for what I have here. And so what I'm going to do is just slowly go through the study questions, and I'm just we'll take a look at the ones that you want to talk about. If no one has anything they want to talk about, that's fine. You don't want to, you can ask me later about it. All of that's okay. Just let me know what you want to zoom in on and focus on, because I know for everybody it's a little bit different, right, in terms of their gaps in knowledge and all that. So here we'll look at this one here first. So this is topic 11, of course, and any questions there, or anyone skipping ahead? They can take a, you can give me anything right now, throw it in the chat, or turn your microphone on. Doesn't matter. Either way is fine with me. I see somebody talking, but I can't hear you. So hold on, let me just check my settings. I wonder if I messed up on my end here. I don't think I have it in my computer muted, but let me just take a look. Try again.
Hi, can you hear me?
I can hear you. I think I had my computer muted.
I just wanted to know how we should efficiently plan for the table with the pathogen, the disease,
Yeah.
the infection site, because there's just a lot, like when you look at the last slide, like Staphylococcus aureus, how do we narrow down like getting to the table information when we've talked about so much?
Yeah, I think there's 2 things to think about. One is a lot of these pathogens are known for one thing, right? So like, Lyme disease, right? So you learn the basics about Lyme disease, right? So it's caused by bacteria, the name of it, what the vector is, maybe some of the basic symptoms. And, for every organism, usually there's some miscellaneous things to throw in there. So Lyme disease, for example, the miscellaneous thing is that it's a vector-borne disease spread by a tick. So many of the infections, that's the thing. They have sort of one thing to say about them, the disease, but then there's a handful there. So Staphylococcus, E. coli, Streptococcus. Just trying to think if there's any others really to throw onto that list that are known to cause multiple types of infections. And those are the ones that, are going to have their own sort of special study category where you want to learn a lot more about them. I don't know, does that help? I know.
Yeah, I think, when I'm going through notes, is it easier to look at, if it's called by its name, work from there instead of picking like the pathogen and then trying to go back and figure out what the name is, or is it a little bit of both?
Yeah, a little bit of both, right? Like some of
Okay.
these are a bit more known by the pathogen name, and then others more by the disease name. And unfortunately, that's just kind of the way it is.
Yeah. Okay. Thank
Yeah.
you.
All right. Somebody is saying, how much should we know about toxins like exotoxins or enterotoxins? Okay, so good question. You definitely want to know the definition of a toxin, right? So you know that it's a molecule and not a pathogen. So it's something that doesn't grow, it's not infectious, but it can cause disease. You definitely want to know the difference between the exotoxin, endotoxin, and enterotoxin. So, for example, an exotoxin is secreted usually by a gram-positive. An endotoxin is usually a part of an organism and often is referring to part of the gram-negative cell membrane. And then, like I said, know some examples of some of the toxins that we talked about. There's at least 6 or 7 that we've talked about that I can think of. Probably some we talked about a bit more than the others, like Botox and the tetanus toxin. I think we talked about the most. So always good to know at least 2 or 3 examples of things if you can study that. All right, keep the questions coming. Or bring them out on Friday if you decide to come on Friday too. And Friday, I'm going to cover most of the same content. So like I said, you can pick and choose which one you come to, one or both or none, or doesn't matter.
Position. There was like,
Sorry, I heard half a question, I think.
yeah, do we need to know the composition of the gut bacteria? There was like 5 different names,
No, I would not worry about that. There's a bunch of organisms in there that are not important as pathogens. I tried to highlight the ones on the list that were pathogens, and those are the ones hopefully you're studying already. So someone says, if you eat a deer with Lyme disease, would you get Lyme disease? That's a good question. Probably not, because if you think about it, we actually eat a lot of organisms that have pathogens in them, but there's a few things going on. One is the pathogen is going through your digestive tract. So the bacteria for Lyme disease has to get in your blood or something like that. It will get destroyed in the digestive tract. And 2, hopefully you're probably cooking most of your meat, and that's going to kill the pathogens as well, right? And so probably not a big deal. But interesting question. Okay, next one says, should we know the different types of penicillin like methicillin and so on? No, do not worry about that. Just know what penicillin does in general, that group of drugs. Somebody else is asking, do we need to know a lot about the contributing factors to disease emergence? So I think you're asking about the emerging diseases section of topic 12, I think that is. So for that, there's kind of, again, it's a list of a bunch of things that can contribute to disease emergence or reemergence. It's the kind of thing, there's a lot there, but know a few examples, right? Some of them we talked about a lot more in class. We talked about, for example, antigenic shift, right? As being how flu can come up with new strains. So that's something we talked a little bit more about. So definitely you want to learn a bit more about that. But it's kind of like I said, know a few examples. That's probably good enough. Anything we focused a lot on, you do want to focus on more. Somebody's asking, will the recording be posted on Moodle? Yes, I'm going to post it on Moodle when I get a chance. So probably this afternoon. Somebody's asking if there will be 1 case study on the final or 2. Okay, yes, good question. So there will be 3 case studies. You'll have to answer 2. So like the midterm, you've got a choice, but there will be 2 for you to answer. It's really very similar to 2 midterms in terms of the length. Exactly double the number of marks and all that. All right, keep the questions coming. Someone says, how important is it to know the things that I have in topic 11 about MRSA? You definitely need to know about MRSA. I know there's a lot of facts and details there about it. Don't worry about statistics and things like that, but you should know what MRSA is. It's multi-drug resistant or methicillin resistant Staphylococcus aureus. So you should know the acronym and why it's important, right? And maybe some of the things that you might do if there's a patient suspected with MRSA, what might you do in hospital, preventative measures and things like that. And then everything else is just general Staphylococcus knowledge, what diseases and things like that might cause. Okay. Someone's asking, what will we need to know about the mechanisms of pathogenicity? Okay, lots. Again, there's another area where it's important to know examples. So there were, I can't remember, it was A to G. So we had the number of organisms, attachment. So each of those things, I would recommend that you know a couple of examples from each of those things and what those things are and what they mean, right? Most of them are obvious, right? Attachment mechanisms, right? So we talked about like pili and hooks and suckers on worms and things like that. So definitely try to know a couple of examples from each of those. Again, you don't need to know statistics like exactly the number of infectious particles for norovirus. That's not important. The concept is more important, knowing that smaller number of infectious particles means the disease is more virulent. Somebody's asking an example of a mechanical vector. So mechanical vectors, so there's two types of vectors, right? The biological vectors. So that usually is an insect or a tick or something that's essential for the disease propagating itself. So for example, in malaria, the mosquito is the biological vector. It's part of the life cycle of the pathogen. A mechanical vector is a little bit more accidental. So in many cases, mechanical vectors are associated with sanitation issues. So for example, I think I showed you a picture in the slides of a fly or a cockroach or something like that, and it was walking across some feces, some fecal matter, and then it got onto somebody's hamburger. So that's a mechanical vector. Maybe what I'll do is I'll move to the next slide here, see if there's anything else in there that anyone wants me to talk about. But keep the questions coming. These are just the study questions that are found at the end of the units. So there shouldn't be anything unique there that you haven't seen before. Just trying to put them up in one place for people to see so that they can ask me about them. Do we need to know about the events in phagocytosis? No. But you do need to know what phagocytosis is, right? So be able to define it or talk about it and those kind of things, right? So, but you don't need to know the individual steps. Good question. All right, keep the questions coming. I'll leave this one up for another minute. If there's no more questions, I'll move to the next one. Somebody's asking about the stages of disease. I think I've got a slide in there somewhere that talks about that. So you've got the infection and then the incubation period. So the incubation period is usually, it means how long the pathogen is multiplying and before you get symptoms. So that could be 1 day, it could be 14 days, kind of depends on the disease. Then you got the symptomatic stage where you have symptoms, and then kind of the getting better, or I guess dying stage, at the end. That's basically what it's all about. So again, it's mostly just being able to use the terminology to discuss these infections. All right, let's see what's on the next one. So innate immunity. So for innate immunity, I know there were lots of skin things. Again, know some examples. It's always good to know examples. I know you can't necessarily learn everything, but examples are always good. And then at least, that kind of gets you started with answering the questions. Anything on that one? All right, I'll move on to part 2, adaptive immunity. I think we covered this quite a bit. I'm just looking to see if there's anything else there. I know there's a definition there for adaptive immunity epitope oops, there it is. I don't think I said that word in class, but it is it's basically part of the antigen that the antibody binds. I won't use that word on the test. A couple of questions here. It says here, will we have to match pathogen names to pictures of them? Nope. You did that in the midterm. I'm not going to ask you that question again. And if I did, it would be one multiple choice question maximum. Next question, it says, is the true and false included in the 40 multiple choice or the matching? Probably included in the true and false. I really can't remember. I have to look and see what I did last year. It will add up to 80 marks is what the exam will be. And I kind of work, usually what I do is I take about 2 or 3 multiple choice true and false set questions from every unit. And that usually adds up to about 40. Someone says, can you give an example what kind of questions will be in the matching, like previous matching were gram stains, microscopes, definitions, often definitions, right? In this case here, you can imagine the last few units, we covered quite a few things, right? For example, diseases, so I could have a matching, the organism with the disease name, or, thinking about the, I feel like I had a question last year that was about the mechanisms of pathogenicity. I'm trying to remember what it was. But, there's lots of things that have examples. So, that could be the kind of thing, on one side, give an example of, let's say a virulence factor involved in attachment to a host cell, and then on the right side, you would select pili or something like that. Sorry, I can't be more helpful, but those I think that's what I'd be thinking of anyway. So any questions about adaptive immunity? I think, like I said, we covered this one in quite a bit of detail just earlier. All right, what about part 3, immune disorders? Again, we covered this a little bit, I guess. I think there actually might be a question or two missing here from this slide, but you can take a look at the study questions yourself with the notes. The last one here, it says here, here's some other study questions for the diseases. I tried to question 7 here or question 7 on the previous one. So this one here, why we do not have an effective vaccine for HIV, mainly because it mutates like crazy. It mutates really fast. And so we could make a vaccine, but then which vaccine do we make? If you think about all the people infected on the planet, there's going to be many, many variants of HIV on the planet. So there's not going to be one vaccine that can cover everybody. Never mind, there's been studies where they did make a vaccine, but it didn't even have an immune response against all of the virus variants in the one individual. So mutation is the answer. Mutates way, way too fast, HIV. Do I accept bribes? Funny, funny, funny. All right, here's the study questions for the last topic, 14. Anything in there you want me to talk about? Like I said, I don't have to cover these study questions. If there's something else that's on your mind, let me know. I can always dig up some answers for you. So hopefully by now, everyone knows the answer to this one here. Oops, not that one. This one here is antigenic drift or shift? Drift. Shift is the fast one, drift is the slow one. So I guess to answer that previous question with the HIV vaccine, it's antigenic drift on a massive scale, really, really fast. All right, somebody wants to know if there'll be a bonus question. Yeah, there'll be a bonus question. Somebody's asking, can I go over number 9? distinguish between meningitis and encephalitis. Okay. They have some overlapping symptoms. Both are dealing with the brain. Meningitis is an infection of the membrane surrounding the brain and spinal cords. Encephalitis is a swelling of the brain. They have some different causes as well. Both of them have multiple causes. So meningitis, a big cause is, of course, Neisseria meningitidis. That's the one we get the vaccine for, associated with saliva and other things. And but also can be just trying to think, 60% of the cases are that and two others. So Staphylococcus, I think, and the one that we never talked about, Haemophilus influenza. So many causes, often bacterial, right? Encephalitis, many causes that probably a few more viral causes, but there are bacterial causes as well. So West Nile virus is a good example where sometimes it's actually called West Nile encephalitis when they're talking about encephalitis induced by West Nile. I think mumps can cause encephalitis, mumps virus. What else am I thinking about? There's a bunch of viral diseases that we never talked about that cause encephalitis. You know, both of them, you can imagine, because the causes are different, sometimes we're looking at different treatments or different preventative features. So meningitis, we do have the vaccine for the Neisseria organism. Encephalitis, in many cases, you're dealing with the brain swelling, which is, you know, giving people something to kind of, you know, like steroids, which will chill the immune system a little bit. So that's kind of the big thing there. Know the definitions, couple causes. I know we didn't cover them in really a lot of detail, so probably that will be in other courses. Unfortunately, we just can't cover everything in one semester, can we? Okay, any more questions? Like I said, this is early in the week. I know lots of people are just barely begun studying, so you can always visit me later in the week. Unfortunately, our exam is 9 a.m. Monday. I'm not going to have office hours at 7 a.m., but I do answer emails, usually during exam times as well, to try to cover any last minute burning questions. So somebody's saying, will there be many questions about latent and active tuberculosis? Well, I mean, I'm not going to have many questions about it, but that's definitely a place to have a good question about it. You know, it could be a short answer or something like that. Usually I'm not going to, usually I don't have open-ended questions like number 8, right? Just, you know, describe the 2 types of tuberculosis, because then you're going to have to make a giant table and you know, people are going to go off in different directions. Usually that kind of thing, I'll be a little bit more specific about asking you the question. So I might say, you know, what kind of symptoms does someone with latent tuberculosis have? Right. So that's very specific. In that case, the answer is they don't have any apparent symptoms. Or I might say, you know, question, you know, what would the X-ray look like on latent tuberculosis versus active tuberculosis? Right. So, you know, something more pointed, right? But I'm not going to ask you a whole bunch of questions, 1 or 2 kind of max about that. But it's worth it to learn about tuberculosis, because I will have overall around tuberculosis a few questions about it. Okay, any other questions? Well, if that's it, I guess this is the end. I showed you my, not my cartoon, but Calvin Hobbes' cartoon. I changed 1 word on that to show you the microbiology. And unfortunately, yeah, you guys have a lot of work ahead of you. But hopefully your studying goes well. Like I said, you can drop in or email me anytime, or you can set up an appointment or a Zoom appointment, and I can certainly answer your questions. Other than that, I guess if I don't see you before then, I'll see you at the final exam. And hope you have a good Wednesday, and and best of luck with your studying. So I'm going to end the meeting now. We will see you later.